Hello everyone, my name is Verna and today I'm going to present paper is phosphorylated-DNA bacterial dye, which is reduce our S level in C. elegans and improving locomotions and longevity. So firstly, I want to introduce you what is phosphorylated-DNA. It's non-bridging oxygen in the phosphate link and is replaced by the sulfur atoms. And this DNA phosphorylation is widely distributed in the human gut microbiome. So it is in the bacteria. And in this work, the PT diet effects on nematodes is studied with some PT-bioengineering bacteria. So to study the effects of the PT on nematodes, they are embedding the plasmid of the gene into the bacteria and feed the worms. So and they find that the RS level decreasing by 20% to 50%, and the age relative lipofuscin accumulations is reduced by 15 to 25%. Moreover, the PT feeding worms are more active at all life period and more resistant to the stress. So they are living longer and their lifespan is prolonged by 21.7%. They also do RNA-seq analysis to study the genes changes compared to the PT diet and non-cheat diet worms. And they find that some longevity genes such as GNK1 and stress response genes like SOD3, GST group, and also T-short proteins, they are changing. And both the Gene Ontology (GO) study and also KEGG enrichment analysis suggests that the neuron activity pathway is upregulated. And there's also some others pathway like DNA repair pathway. They are downregulated in the good appetite young worms. And these findings altogether saying that the PT bacteria has the poor longevity effects. So we will come to their result study. So the first figure is about PT containing bacterial food reduce RS level in C. elegans. So to compare the RS level and lipofuscin accumulation of C. elegans fed with E. coli, OP50 or the blue script based plasmid bacteria. So there are two bacteria. One is control one and one is fed with the plasmid genes D and D, which is a cluster from the Salmonella bacteria and no more E. coli OP50, which is a negative control. So we can see the two groups is one is S minus and one is S plus. S plus is the plasmid containing bacteria and S minus is the control. We can see that compared to the control, the RS level is significantly decreasing. But it's they have different time points. So they check the RS level at day 1, day 4, day 8, and day 12. The reason why, because they want to examine the long-term effects of the of this antioxidative effects. So they find that the RS level decreased by 18.5 and 19.1 at day 1, and day 4 is decreasing by 19.1, and day 8 is decreasing by 32.7. But come to day 12 is slightly decreasing, only 0.9%. And likewise, they use a different diet. Different fluorescence probe, which is DCM. So why they use DCM? Because this probe is more sensitive to the hydrogen peroxide. So they use this probe to check the RS level, and they find that it decreased by 19.9% at day 1 and 54% at day 4 and 21% at day 8. But there is no significant change at day 12. So we can say the RS suppression by the PT containing bacterial dye is in good agreement with the previous study of the antioxidant property of this PT. So I'll come to figure 1C. We can see it shows that age accumulation of the age pigment, which is lipofuscins, is also changing, right? We can see that lipofuscin accumulation of these worms fed with PT containing bacteria is slowly accumulated than the normal OP50 diet. So they delay the rate of the aging. They delay the rate of the accumulation of the lipofuscins through the life. And come to figure 2. They also check the phenotype changes potentially caused by PT containing food. So they track the locomotions and lifespan and body and the atlays of the C. elegans using the wild type strain. Comparing to the PT containing bacterial food and the normal OP50 food. So you say figure 2A is they checking percentage of the movements. So the worms fed with PT containing bacteria is more active. Probably is particularly during day 10 to day 16. Day 10 to day 16, so the motility is increasing. And figure 2B, they're checking the lifespan. The lifespan of the animals, the C. elegans fed with D&D, as plus E. coli is significantly longer than the control one. So which means the PT diet is increasing the lifespan as well. So I'm coming to figure 2C. They also check the body size is not changing, and also they check the outlay is not changing as well. So they do not the PD containing food is not interference the body development. And then we come to figure 3. So figure 3 A to D, they want to check whether the stress responses is PT containing diet, whether it's also affecting the impact the stress response. So figure 3A, they track paragraph is not having, it's actually increasing the survivals. The mean lifespan of the paragrams is increasing from is increasing significantly with the paragram treatments. The lifespan p-value is smaller than 0.05, and also with the CR treatments, CR progeny treatments is also oxidants, so it also increased significantly. So PT containing diet, it help in the case of paragraph and CR progeny treatments. And so it also against the heat shock. So it also rescued the survivals under heat shock significantly, but it doesn't have any it cannot rescue the survival under exposure to the ultraviolet radiations, even for the long-term S plus diet. So maybe there is some the fat going there. So and they track its antioxidant properties, and then they come into checking the RNA-seq to see the sequencing results. So we can show in figure 4 A and E. So A, and so each groups of the samples is having really high quality FPKM. In RNA-seq, we can see it's really consistent, and PT feeding groups presents relatively high FPKM values. So in figure 4B, we can see that compared to the non-PT fat nematodes, more genes are transcribed in those PT fat diet. And the number of the differentiate expressed genes in the D4 samples is more than the day 12 samples. And because we think they think the day 12 sample is not the effect of the PT is smaller. It's really smaller because it's getting really aged and old worms, and the most affected day is day 4. And also they find that the biological functions of the Qiang and downregulate genes point to many relative enzymes and the lifespan relative DEF2 and DEF16 genes. So in the recently study of the mitochondria longevity regulated by ER genes, there are many transcripts are changing as well. So this is their transcriptome results. And come to the analysis of the type of genes, the group of gene changings, which is more clear. So they for the transcriptome they also do the clustering to show the gene expression related to stress and stress response and agings. So from A to H, we can see that SOD3, SOD5, and they are increased by 10.3. Sorry, fold change. We can see the clustering. So we see the RS genes. They are actually compared to the S minus and S plus. Some of the genes are increasing with the S plus. For the heat shot, we see that some of the genes are decreasing at day 12 compared to the day 12 control. And also see the GST genes, they are actually even at day 12 and day 4 it's not, they are also increasing a lot at day 12 and at day 4 compared to the control. So which indicate that the long-term intake of the PT containing food is changing the sulfur metabolism in the nematode and induce the expression of the GST. It lead to the life extension by increasing antioxidant activity, because GST group is antioxidant activity, having antioxidant activity. And for the qPCR, they also check the expression of these RNA-seq results. So we can say that PT containing bacterial food, it acts as the antioxidant directly to oxidative stress, and change many gene expression by involvement of the nematodes transcriptome network. And interestingly, there are some like antioxidant enzymes such as SOD. We can see SOD is here is increasing when PT diet is fed to the worms. But at day 12 it's also increasing a bit, but it's not very much. And also check GNK1 is increasing with the PT diet. And GST, so GNK1 and GST group also increasing. So interestingly, they focus on SOD and GST groups as the main genes. So come to figure 6. Checking the global Gene Ontology, GO, and enrichment, and also KGG. It shows that GO enrichment of the difference between transcriptomes of the C. elegans S plus and S minus samples. Also, overall, the categories are enriched in cellular components. Biological process and molecular functions. We can see there are some GO groups that is then here. So the cell cycle process is decreasing, and protein's phosphorylation is decreasing. For the KEGG pathway enrichment of the PT feeding elegans, in figure 7, among the top 3 regulated pathway, a DNA repair and mismatch repair and this pathway are downregulated, but the neuron-active ligands receptor interactions are regulated. As showing with figure B to C, you see that the most dramatic change in the top rank regulation pathway is the range of 2 to 3, 4. 2 to 3, 4. And the top one is some neuroactive ligands receptor interactions and calcium pathway, calcium signaling pathway, and purines metabolism and drug metabolism pathway. But really interestingly is the glucosamine metabolism is upregulated in D4 samples, and many GST genes are induced, which is consistent to the differential gene expression in figure 4. So this stress that PT containing worms is changing the sulfur related metabolism in organism. So when it comes to discuss the main investigation of the paper, they first basically the overall PT containing bacteria food effect of the C. elegans is illustrated in figure 8. So basically they have the DND plus plasmic bacteria, which brought the unit phosphorothioate materials into the worm's intestine tract. And the worms and the bacteria is digested into many small nutrients molecules, including the phosphorothioate and their nucleotide oligomers. So the long-term intake of this diet, it can help the worms to maintain a relatively low level of RS stress in their body and enabling their higher locomotion and extending the lifespan. So that's the main finding of this paper. So and there's also giving some superior phenotypes that could contribute to complicate regulation of the genes expressions. So that's the main points of this paper, and also welcome to ask me any questions in the comments, and I will answer next week. Thank you.